Oh, could you speak a little louder?
Oh yeah, sorry.
That's okay.
I'm Marianne, and this is what I will be discussing today. Beating cardiac cell and pacemaker macrostructures produced on 3D printed substrates. So, just the abstract real quick. The human heart's electronically responsive muscle tissue can now be extracted, expanded in culture, and returned to the patient as therapy. However, the scale of current autologous transplanted tissue remains microscopic, and the pacemaker tissue is lacking. So this project is aimed to produce functional macroscopic beating tissue that contains its own pacemaker, as you will see in the following slides. Just a background on the human's heart's ability to repair. The heart is the hardest, most vital working muscle in the body. Unlike skeletal muscle, which can regrow throughout exercise at any age, the heart is limited to a regeneration rate of 1%. So, loss of heart function due to trauma or tissue loss can affect the heart at any age. And so my experiment today is to produce or to further delve into the ability of a cardiac cell to beat in culture and to examine the conditions to which it beats. So this is other techniques for regeneration of heart cells. So a non-invasive alternative for augmenting the number of beating heart cells is to take a small piece of the heart and to expand it outside of the body and return it to the patient. However, although this approach has yielded some astonishing results, they don't necessarily have the ability to repair gaps in the heart created by tissue destroyed during the heart attacks. Hypothesis. My hypothesis is to extend the techniques in producing beating cardiac cultures. To do this, I hypothesize that we design a 3D printed mold that can house a gel containing organized beating cardiac macrostructures. In this study, the necessity of the cells to adhere to a substrate is explored using controlled substrate shapes, which is printed through a 3D printer. If successful, this method described here can lead to the ability of regeneration of functional cardiac tissue that can be transplanted to humans. And this can be accomplished by growing cardiac cells in laboratory that are able to attach to a body's natural scar tissue matrix material, which is fibrin. And controlling the shape in which the fibrin conducts beating movements, and using the cells grown on the matrix to install patches of tissue for repairing larger damaged areas of the heart. So, starting off with the first video, it's called Applesauce, and it's a structure of adherent cardiac cells beating in culture. So this was the starting point of the experiment. And as you'll see in future videos, the contracting of the cells. And this is 2 beats. So there was an identification of the 2 beating centers, one up there on the right and one down here. And this video was significant in the laboratory because there was 2 beating centers in it, so it showed the potential for several pacemakers. which can be further explored, which you will see in the further experiment. So we started off with the control experiment like you saw with applesauce and 2 beets, and we thought of a fibrin matrix overlay. So there's already beating cardiac cells, and we thought to enhance the beating by spreading a layer of fibrin on top. So we started with the control, and this time we added more fibrin on top. And so we saw that the cells migrated from the previously beating culture to the fibrin on top. So as you will see with the next video, you get this macro structure that is beating in the okay, so this we call iguana. So as you can compare with the previous videos, this iguana is beating more rigorously, and so yeah. And then it was actually to the point where you can see it with your naked eye. So the structure was suspended in the matrix, and it was so large that you can look at the culture plate and see it with your eye. And these are the cells actually on the fibrin strand. So you can see how it contracts in that beating center. So controlling cardiac beating on 3D printed matrix. We sought to build a macroscopic beating structure with the use of modern technology, which is 3D printing. So we thought of making little boats to house the fibrin and the cells. So this mold worked much better for our needs, as it was able to hold more matrix than in just the circular culture. And we used it, and so it was a cast, so we then removed it, as you will see in the future slides. So these were the steps. First, create a matrix consisted of gelatin, fibrinogen, and the envelope. Put the matrix in the boat, add thrombin to polymerize the material, so fibrinogen and thrombin mix together to form fibrin. We remove the matrix from the boat into a cell strainer and immerse the cell strainer into a culture dish for differentiation. There were 3 types of media to cook to as a type of to see how each one affects the culture. So there's the content in each media. So the change of media was twice a week, and this was simply through trial and error to see when these cells beat. So we did it twice a week, and we saw large macrostructures of fibrin containing beating cardiac cells created in all 3 types of media. So due to the thickness, I don't know if you're able to see that structure, but that's what we saw in the streamers. So the cardiac macrostructures were able to continue growing and attach themselves onto the cell strainer, and the attachment of these cells to the strainer enhanced their beating. So in conclusion, cardiac macrostructures can be produced easily in vitro the laboratory, generating a beading structure capable of being manipulated by a surgeon is within reach. And that was our goal for this experiment. Controlling the polarity of beading and organization of the resulting tissue by the combination of the traditional cells culture and 3D printing allows the cells to self-organize as directionally beading tissue. We hope to enhance this project by improving the 3D printing process by injecting binary matrix components, factors, and shape guides simultaneously. And addition of blood vessel differentiation factors to the matrix is also desirable. And the beading tissue, pacemaker properties, and vascular features could be prepared in lab in advance of autologous transplantation into the patients. And to say thank you, I've included a heart shape we found in the culture.
Quickly with the questions, yes?
Is there any data on how much, let's say, per square inch or millimeter pulse generated by these beads?
We haven't. For this experiment, our purpose was to see the different factors in which the beading occurred, and so that would be in the future when we solidified our data on what factors and how much of the factors we need to produce the beading structures.
All through the presentation, you're talking about the 3D printed substrates. You didn't really see where you developed those 3D substrates.
Through the, do you mean the structures?
How did you print them to make these 3D?
Okay, sorry, it was part of the presentation, but I must have overlooked it. We used a previously generated template for the 3D printer in which we have, and we used that and we modified that to make the small boats in which the cells were in.
So was this your project? And are you working in a medical lab or?
The laboratory is actually a microbiology at a local community college in which I'm from. But our idea, or my idea, was the 3D printed structures, and it was thought about from previous beading structures, but I modified it. Sorry, I modified it into incorporating the 3D printed structures.
Okay, so the actual application of it. Okay.
You have a question?
Can you create an artificial heart with this microstructure using 3D?
I'm not certified in any way to do that.
But it can be created, I mean.
Theoretically, it
It's
can.
scaffold, yes. It's more complicated, yeah.
Lots of tissue types.
But that's possible project, yes, for everybody to be involved, yeah. Scaffolding, it's another story. Anyway, and no questions, we better move, yeah? Thanks again, and we have 2 more presentations, so we should be done by midnight, yes?